Dear students, today I am going to discuss about autoimmune diseases and transplantation rejection. I already discussed in previous classes what is autoimmunity. In autoimmunity, what happens is like our body will not be able to distinguish between self-antigens, that means antigens from our body, and foreign antigens, that means non-self antigens. Okay, and ultimately that results in production of auto antibodies. The antibodies which are produced against foreign bodies are antibodies, but if it is produced against self antigens, it's called auto antibodies. So that results in autoimmune reaction or autoimmune disorder or diseases. Okay, another term you have to be familiar is immune tolerance. Okay, in which our body should be, our immune system is tolerant against autoimmune reactions. Okay, that means our body is able to recognize a self antigen. Okay, and it will not react against self antigen. Okay, so that is immune tolerance. So if immune tolerance is disrupted, ultimately it will result in autoimmune reactions. So how this disruption of autoimmunity, disruption of immune tolerance occurs, or what is the mechanisms behind the occurrence of autoimmunity, that we are going to discuss now. These are the three important mechanisms. One is sequestration of autoantigens is disrupted. Okay, so whenever there is complexation or sequestration of autoantigens, they will be complexed and they will be removed from the body. But if there is any problem in this complexation, that will result in autoimmune reactions. Another is problem in generation and maintenance of immune tolerance. Immune tolerance is also called energy. Okay, if there is any problem in that due to problem in the mechanism in which T and B cells function, then that will result in autoimmunity. And also any other regulatory mechanisms that is involved in immune system which disrupts the immune system may trigger autoimmune reactions. And the mechanisms or theories of autoimmunity may be due to exogenous or endogenous factor. If it is exogenous, means it's from outside, and endogenous from inside the body. Okay, and this may be broadly classified into three, that is either immunologic, genetic or microbial factors. First we will discuss about immunologic factors. Any failure of immunological mechanisms of tolerance will result in autoimmunity. The best example is polyclonal activation of B cells. That means some problem in the immune tolerance which result in multiplication of B cells, resulting in different clones of B cells. So once these clones are generated, these clones may be acting against each other, reacting against each other. That is self-reacting B cell clones. Okay, so that will result in autoimmunity. Another is decreased T suppressor cell activity. T suppressor cells are actually cytotoxic, and they will remove toxic cells, okay, unwanted cells. Okay, if that activity is disrupted, and along with that if there is increased T helper cell activity, that will result in autoimmune reactions. Another is fluctuation in anti-idiotype network control. This network control occurs between antibodies which have some similarity in their variable regions. For example, antibody A and B are having some variable regions and they have some similarity in their variable regions. Okay, they are called as idiotypes. Okay, there may be chances that these idiotypes may react against each other, but that is prevented by anti-idiotype network control. Okay, if there is any fluctuation in that, that may result in autoimmune reactions. And if there is any problem in the sequestered antigen released from tissues, okay, that means we already discussed self antigens may be complexed and they will be released from the tissues to the tissues, and ultimately they will be removed. But if there is any problem in that, okay, that this antigen may accumulate, self antigen may accumulate, triggering autoimmune reactions. That is explained here. Next is the genetic factors. Genetic factors, for example, if there is overexpression of class 2 HLA antigens on tissues, involved in autoimmunity that may result in autoimmune reactions. And that is usually accumulated in the family, because if the parents are having that problem, that may accumulate in the children also. Another thing that triggers autoimmunity is microbial factors. For example, if a person is having genetic and immunological problems, as we discussed now, okay, and if there is any microbial trigger, okay, that may there are chances that may end up in the generation of pathogenesis of autoimmune diseases. For example, especially if there are viral infections, for example, Epstein-Barr viral infections, there are chances for that. Next is classification of autoimmune diseases. The autoimmune diseases may be broadly classified as organ specific or localized, and non-organ specific or systemic diseases. In the organ specific one, what happens is like the autoimmune reactions are targeting a particular organ only, but in the other case that may affect any organ, although the starting point may be a particular tissue, it may spread to other areas also. In the case of organ specific, best example is thyroid gland, in which there is autoimmune hyperthyroidism or hypothyroidism that will result in symptoms in thyroid gland related to thyroid gland only. Another is type 1 diabetes mellitus, in which the main theory for type 1 diabetes is autoimmune destruction of cells of the islets of Langerhans. Okay, that will be affecting the pancreas only mainly. Okay, so that is actually organ specific or localized diseases. And others are the autoimmune destruction of blood cells. Now, organ non-specific or systemic diseases, the best example is some collagen diseases. The best example is rheumatoid arthritis, in which it begins in the joint space first, but the autoantibodies produced may circulate all through the body through blood, and it may affect other organs also. This is a classification of the autoimmune diseases. It may be organ specific or systemic. Another is like the autoimmune reactions may either, the main mechanism may be the generation of autoantibodies, or sometimes this may be mediated by T cells. Okay, so these are classified as like this, and you can write one or two examples from each type. Okay, and that is explained in the coming slides. Okay, next is which diseases which are postulated to be autoimmune. Like the exact reason for these diseases are not known, but it is supposed that the main reason for autoimmune, the main reasons is maybe autoimmune. So that is why this is explained as diseases postulated to be autoimmune. And the best example is inflammatory bowel disease. That is a disease that is affecting the small intestine and more often large intestine. Okay, so it is mainly confined to the large intestine, and the theory behind it is like it is supposed to be autoimmune. Okay, so that is it. I will discuss some diseases in detail. Myasthenia gravis, it is an organ-specific disease that is affecting the skeletal muscles. Autoantibodies are produced against skeletal muscles. Rheumatic fever occurs when antibodies are produced against streptococcal infection, and that may damage the cardiac muscles. Okay. Next is autoimmune diseases of blood cells, that is hemolytic anemia, then autoimmune thrombocytopenia. All these are examples. Next, multiple sclerosis is affecting the nerves of the CNS, in which the myelin sheath of the nerves are destroyed, resulting in nerve damage, and that disrupts the communication between brain and body and results in spasm of extremities. Okay. Another is Graves' disease, that is autoimmune disease that is affecting the thyroid gland, in which autoantibodies are produced against thyroid gland. Next, non-organ specific include SLE, that may affect any part of the body. Another is systemic sclerosis. I already discussed multiple sclerosis. Multiple sclerosis is actually affecting the CNS only, but here systemic sclerosis or scleroderma may affect any part of the body, but the main areas affected are the skin and small blood vessels. Rheumatoid arthritis I already discussed. It may start in the primary joints, but mainly in the joints, but may involve extra articular tissues like skin, blood vessels, lungs and heart. Next is Sjogren's disease. It is basically the autoimmunity triggered dryness of eyes and mouth, and it is due to the destruction of salivary glands and lacrimal tear glands and all those things, and later it may affect other organs also. Next, now we will move to the transplantation rejection part. I already discussed about transplantation while I was discussing about MHC. Now we will study about that in detail. So the different types of transplantation grafts are discussed here. Autografts are grafts in which the donor and recipient is the same individual. That means tissue from one part of the body is taken and maybe put in another area. Okay, one example is that it is autograft, and here the chance of rejection is least. Next is isograft, in which the donor and recipient are of the same genotype. Here also less chance of rejection. But allograft, in which the donor is of the same species but of a different genotype. Here there are chances of rejection. That is usually happening in transplantation. Okay. So and the most rejection happens in xenograft, and that is usually not happening nowadays. Now, okay. So now we will discuss about rejection, how the transplantation rejection or allograft rejection happens. It is a complex process that is triggered by the two pathways: direct pathway for cell-mediated immune reactions and the indirect pathway. The main pathway is direct pathway, in which the T cells, T lymphocytes are involved. Okay. Now we will discuss about the direct pathway or cell-mediated immune reactions. Here the main cells involved are CD4 and CD8 T lymphocytes. Okay. Then the cells, T cells of the recipient. For example, if I am the recipient, my T cells will recognize the donor MHC molecules on the surface of the antigen presenting cells of the graft. Okay, I already discussed about MHC by MHC, and I showed you the diagram of antigen presenting cells with MHC and how it is getting it is received by the T cells. All these things I showed in the picture. So here the same thing happens with my T cells. I am the recipient, will recognize donor MHC as some foreign body, okay, and will start attacking. Okay, so CD8 cells will recognize donor class 1 MHC, and it will mature into cytotoxic T lymphocytes that kill cells in graft tissue. Next is CD4 T cells recognize class 2 MHC, and these two reactions will result in cytokine production, inflammatory response, and accumulation and activation of macrophages, resulting in graft injury. Next is the indirect pathway. Okay, here the antibodies, alloantigens are produced against alloantigens, and this is a delayed type for hypersensitivity reactions. Delayed type of hypersensitivity reaction is a type 4 hypersensitivity reaction. Here CD4 T cells are also involved. Okay, this is a continuation of the direct pathway. Okay, I already discussed that. Then what happens is, like, there may be chances of the production of antibodies once T cells, host T cells, that means if I am the recipient, my B cells, okay, sorry, not T cells, B cells will recognize the alloantigens and produce alloantibodies to produce antibody response. Now the types of rejection reactions, it may be hyperacute, acute and chronic. If it's hyperacute, the reaction begins within minutes to hours, but nowadays that is not happening usually, because cross-matching of donors, lymphocytes happens before the transplantation, so this is not happening now. Next is the acute rejection, happens within few days to few months of transplantation. Next is like chronic rejection, where it happens over a period of months to a year. For example, in the case of renal transplantation, usually if there is rejection, there will be increased serum creatinine. Okay, but usually in the renal transplantation, if there is chronic rejection, this increase in serum creatinine may be seen only after months or years. Okay, so that is an example of chronic rejection, and the mechanism behind this may be immunologic or ischemic. That means there may be less supply of oxygen to the particular area where transplantation happened. Now the transplantation antigen. So antigen recognition, that means the cross matching of antigens or compatibility of antigens are very important before doing transplantation. Otherwise immune reaction will occur. For example, in the case of blood transfusion, we have to match the ABO and RH antigens. That means whether A positive or B positive, A negative or B negative, that we have to confirm before doing blood transfusion. Likewise, we have to do some compatibility testing before doing transplantation. For example, like MHC molecules should be matched, okay, between the donor and the recipient, okay, as well as other host donor antigen compatibility should be done. Next is amyloidosis. That for that you have to just refer to this YouTube. After that you go through these small notes, okay. Usually a 5-mark question will be asked for amyloidosis, so you can prepare a small note from your own small note from that YouTube video plus this brief description given by me. This is usually asked for a 5-mark question for the university exam, okay. So that's all about this immunity part 5.